One of the most common questions that I often get is, okay, when's the mite going to get here? If it were to be found in California around almond pollination.
In some ways, it's a little bit weaker than Varroa because it does rely on that brood of just the right age.
This is something that reproduces faster than Varroa.
It'll be a lot easier in these cold winter climates.
This is a potential method of dispersal. They're always providing an environment for Tropilaelaps to reproduce.
And that could be an advantage to us because we can manage the brood, right?
And it's just going to be another problem that you're going to deal with.
Hi, everyone. Thank you for joining us again. This is another Betterbee interview with experts where I get to sit down and talk to cool people who are doing cool things that relate to honeybees and to share those conversations with you. So today I have invited on Rogan Tokach, who is a PhD student at Auburn University. He's in Geoff Williams's lab in the entomology and plant pathology department. And so he is studying all sorts of exciting stuff. You know, he's studying Varroa, he's studying honeybees, but he is also studying Tropilaelaps mites. And that is why I have asked him to come and talk to us today, because he is one of the very few people who is going and actually doing research on Tropilaelaps, who is learning what these mites are doing, what threat they might pose to our bees. And so I wanted to get some information straight from the horse's mouth, as it were. No offense, Rogan. And invite him on to talk a little bit about these very interesting and perhaps very scary mites. So thank you so much for coming on.
Yeah, thank you so much for having me.
I guess we should maybe start with the most basic biology here. Like, what is a Tropilaelaps mite? What are these creatures and what are they up to?
So when we're talking about Tropilaelaps mites, there's actually 4 different species of Tropilaelaps mites that can essentially parasitize honeybees. And the one that we're most interested in is Tropilaelaps mercedesii. Now, there is another one that will actually parasitize because these are the only two, there's only 2 that actually go on the western honeybee, which is Apis mellifera. And so Tropilaelaps clariae is one that has really only been found in, I believe, the Philippines. And so they don't really know why it's only staying there. But Tropilaelaps mercedesii is the one that seems to be a little bit more mobile. And it's the one that has kind of spread throughout, especially Southeastern Asia, and is potentially threatening to get into kind of the Eastern Europe and Western Asia areas.
So what regions are these mites currently living in right now? It's mostly Southeast Asia, or what countries are they in, I guess?
So yes, it is primarily Southeast Asia. You're talking about areas like China, Thailand, Cambodia, Indonesia. And then you do have like 1 or 2 countries in Australasia as well, also South Korea. And so the big worry is that they're beginning to spread westward. And so these mites are similar to Varroa in a couple of different ways. And I like to a lot of times start with talking about the similarities to Varroa because it gives beekeepers something that they can generally latch onto and say, okay, this is where I see that they're similar and this is where I see that they're different. So they're parasitic mites. They're going to reproduce fairly similarly to Varroa in that they reproduce within the brood cells while that bee is pupating underneath those cell cappings. And they are capable of vectoring some levels of viruses. They don't have a lot of viral research done, but at least they can vector DWV. Now you start to see a few different differences between them. So Varroa, of course, feeds on larvae, pupae, and adult bees. Tropilaelaps actually doesn't feed on the adult bees. And so they're only feeding on larvae and pupae. Now, also, it seems that Varroa generally actually have kind of a mandatory feeding phase requirement after they emerge as a daughter mite from a cell. They'll usually ride around on a nurse bee, feeding on that nurse bee for somewhere around 5 to 7 days. Tropilaelaps mites don't feed on these bees, adult bees, so they generally actually will then reinfest a cell, usually within 2 days and sometimes even less than that. So what you tend to find is that these mites can coexist within the same colony, but over the course of period of time, usually Tropilaelaps ends up actually out competing Varroa and kind of taking over and becoming the predominant mite species within those colonies when you do see colonies co-infested with both mite species,
Got
just
it.
because they reproduce at a faster rate.
So the mites the Tropilaelaps don't have to stop for lunch like the Varroa do, and so they finish reproducing, they come out and they say, well, I have nothing to do, might as well go reproduce again. So it lets them just
Exactly.
keep growing and growing and growing. And we thought Varroa were bad.
Yeah, and so that's one of the bigger problems is Tropilaelaps mites actually also lay eggs at about 6 hour faster intervals. They're laying about 24 hours compared to the Varroa's 30 hours. And so they just have a few different ways they're actually reproducing a little bit faster. Another interesting thing is that Tropilaelaps, whereas Varroa will just feed on one site on the bee while it's pupating, they'll just constantly feed, open up that same feeding site, feed on it. Tropilaelaps will actually feed on multiple different sites. And there was a really recent publication that came out showing that they are feeding on hemolymph while they're in these cells primarily. I believe that's a Hahn 2024 paper
Mhm.
that Olav Rappel was on, Dr. Rappel. And so it's really interesting to see that there are some similarities, but some pretty stark differences, which allowed Tropilaelaps to kind of subsist and reproduce more quickly.
Geez. So they're running around, they're feeding on multiple sites on this poor developing pupa. Do they have a preference for drone brood, just like Varroa do, or do they not really care what cell they run into?
Yeah, so that's a great question. And interestingly, they have a slight preference based on the literature for drone brood, but it's nowhere near the level of preference that Varroa has. Whereas Varroa is, sometimes 5, 6 times as likely or even more. Tropilaelaps is maybe like just under 2% more likely or
So,
something like
so I
that.
can't just pop in a frame of drone brood, let all the Tropilaelaps run in, and then throw it in my freezer. That's not going to be a great strategy here.
It wouldn't be as an effective strategy as it is for Varroa.
Okay. So they're running around, they're feeding on these bees, they're potentially transmitting viruses, and I've done my share of virological work. It is hard to do, so it's understandable that that's always going to lag behind our understanding of exactly what virus stuff is going on as these things are reproducing. What are the consequences for a beekeeper who has to deal with this? And I guess maybe we should start with the beekeepers who are living in the native range of this mite and are actually trying to keep bees, not as it's invading, but just where it's been for a long time.
Yeah, and so first of all, I want to point out that I like how you use the phrase running across the frames because you don't really
Hmm.
see Varroa actually outside of the cells or outside not on bees. Whereas Tropilaelaps in a heavily infested colony, you will see them actually running along the frames. Tropilaelaps move much faster. They're about a third of the width of Varroa, so they're smaller, they're more difficult to see, a similar length, but they have longer legs, and so they use these legs to rapidly move around the colony, whereas Varroa can kind of more slowly plod along in and out of cells.
Got it. I guess
But
that makes
when,
sense because
yeah,
because Laelaps in Greek mythology was the hunting dog that could catch anything, right? It was the fastest hunting dog or something like that.
I was unaware of that, but that
Well,
could
there
be
you
the
go.
case.
You can put that in your next lecture.
Exactly. So when we're talking about what these beekeepers are doing, you're really seeing that it's a large problem in areas that don't have a natural brood break. And so in areas
Hmm.
that constantly have brood year round, they're always providing an environment for Tropilaelaps to kind of reproduce. And one of the interesting things is that things such as Amitraz and Hopguard, which have been proven to be somewhat effective for varroa mites, especially Amitraz with apivar strips, they don't work as well for Tropilaelaps.
Hmm.
And that's likely because these mites aren't really out on these adult bees. As I said, these feeding phase requirements, they don't have one on adults. So they're going back in and reinfesting into these cells. They're being protected by these cell cappings for a lot of their period of time of their lives.
Right.
And
So
that's
something
why,
something like an apivar strip requires that a bee bumps into it and then gets some of the
mhm.
chemical on their bodies, and then the mite comes in contact with that bee, and then they get sick. But with a Tropilaelaps, they're just not on the bees, so that wouldn't happen.
And so we see beekeepers a lot of times in these environments, they are doing a decent amount of formic acid treatments because, of course, that will kill, that'll penetrate those cell cappings and kill mites within those cell cappings. Another thing that's interesting though is that they have had some success with fluvalinate and flumethrin,
Hmm.
which is a similar kind of method of actually killing the mite, obviously different active ingredient to that of apivar, but you're still putting these strips in the colony. And I don't have a great answer for you on why this seems to work a little bit better than apivar, but we do have one of my colleagues at Auburn University, Dan Arell, who he kind of hypothesized that potentially we were talking about it, and maybe it's because since these kind of are pyrethroids, they're more fast acting kind of acaricides and
Hmm.
insecticides, which are able to kill the mite with a little bit less dosage requirement and not the length of exposure that potentially amatraz is needing.
Got it. And then obviously, fluvalinate, tau fluvalinate is the active ingredient in apistan strips, which we all used, all beekeepers used for Varroa until the Varroa evolved resistance. And the issue there was that the tau fluvalinate was getting into the wax and then it would sort of linger there. And so the Varroa were exposed to low doses and developed resistance. There's also some evidence of damage for developing queens in that contaminated wax. So if we all had to leap back to using apistan strips to fight Tropilaelaps, there would be some downside. It's not like we can just grab something off the shelf that's going to instantly solve our problem.
Yeah, and so it's interesting because these beekeepers that we kind of talked to over in Thailand, been able to kind of speak with some of these commercial beekeepers, and it does seem that, 4 to 6 months out of the year, they are having some form of treatment in these colonies. And they're doing something. And so one of the big problems with Tropilaelaps is it's not something that you can just go out and do an alcohol wash to check your infestation levels. And so what these beekeepers, a lot of them are relying on bald brood within the colony. And so I talked to one beekeeper that said, "Oh, if I go in and I see like 2 frames with over 30 cells of bald brood, then immediately I know that I have kind of a mite infestation problem and I need to get a treatment in
And
these colonies."
bald brood there, just for those that don't know, is going to be pupae who have been uncapped by the workers outside because they've said, oh, there's something going wrong in this cell, better open it up and go in and try to address that. So it's a behavior, it's a trait or a symptom that you can see, but it only works if the bees are hygienic and are sensitive to the smell of a reproducing Tropilaelap mite or a wounded pupa inside, so that they go and open it. So it's an indicator, but it's an indicator that not every bee would have. If you had a bee that was not at all hygienic, they'd just say, oh, there's something weird in that cell, but I'm going to leave it capped over, and the beekeepers wouldn't even know that.
And unfortunately, there's no real threshold to back up when you need a treatment. It's all kind of anecdotal
Hmm.
on, and there's no, even for Varroa, it's just if you have bald brood, you likely have a Varroa mite infestation problem, but it's difficult to kind of understand the level of
Hmm.
infestation.
So how many times have you gone to Thailand now to do this work?
So I was able to go my first year of my PhD the past summer of 2023, and I went for about 2.5 months to conduct a management trial and also look at some potential routes of dispersal, which I hope we can cover a little bit later. And then I was also able to go back over in January for just a little over 2 weeks of 2024. And we were hosting some Apiary Inspectors of America and doing a workshop with them because we kind of thought that, hey, these are the people doing a lot of these APHIS animal plant health inspection service. They're doing a lot of these kind of different monitoring things, and they're the ones doing a lot of monitoring for Tropilaelaps, doing these different tests. And so they're going to be kind of on the front lines of potential first detection.
Right. So you train the frontline soldiers to make sure that they know what they're looking at and aren't, seeing one mite and thinking it's a different kind of mite or something like that.
And then we'll be going and we'll be hosting a workshop in Auburn for the rest of the inspectors that weren't able to visit because we only took a subset.
Hmm.
And then that subset will then help train the rest of the inspectors that didn't actually go over to Thailand. And so we're trying to, it's kind of a train the trainer approach to really try to get as many people as possible educated on AAA and especially those most likely to encounter it.
Excellent, excellent. So in Thailand, the Thai beekeepers that you talk to or that you work with, are they sitting there pulling their hair out over Tropilaelaps, or is it just sort of something that's annoying that they have to deal with, much like I find Varroa annoying, but I deal with it and I'm able to keep my bees alive?
So it's interesting, and it's actually probably more of the latter of those scenarios is they're dealing with it, it's frustrating, but it's not always causing humongous problems. And so I will say though, they do a few things management wise that is really giving them an advantage. One of them being that they keep much smaller colonies over in Thailand. Since it is warm essentially year round, they don't have to worry about building up huge honey stores and then having their bees subsist through the winter. And so they're generally keeping, these colonies at less than probably 7 or 8 frames of bees, and they're all in single boxes. And
Hmm.
usually they're just adding more frames for the honey flow and then removing them. They're not adding extra boxes of supers. So, your beekeepers that are stacking the supers, 3, 4, 5 high, they're not getting to near the level of bees. And so what that allows is it allows for a lot less actual cap brood area within these colonies. So of course, the less cap brood area, the less opportunity that these mites are going to have for brood area to then reproduce in.
Hmm.
So that's generally just a small practice that they do that keeps these mite populations down. But I don't know how feasible that's going to be in the US for a lot of our beekeepers and what we want to accomplish.
Right, it's a very different style of beekeeping.
Mhm.
It's not like we could just have them arrive and have everybody shift over to that Thai style of management and then, not have any consequences. Got it. Okay. So you mentioned dispersal. Maybe we should talk about that. So these mites are living inside, and this is obviously something near and dear to my heart. A lot of my PhD dissertation on Varroa was how do Varroa spread between hives or between colonies. So if I'm a parasite, I get into this colony. It's this beautiful smorgasbord of brood. I can eat and eat and eat. I'll spread all the viruses I want to because I'm going to be fine no matter how many viruses I spread. And at a certain point, I'm going to look around and say, "Uh-oh, things are really taking a turn. There goes the neighborhood." So I want to get myself into a new honeybee colony. How do I do that if I'm a Tropilaelaps mite? What do we know about
Yeah.
it?
And so this is fascinating. And this is something I too am really interested in. I might have to ask you some questions about your thoughts on Varroa dispersal and what you might think these mites are doing. Because the unfortunate thing is we don't know a lot. And so Varroa mites, we know that they will be on the adults, and at some point they have kind of a preference of preferring to be on nurse bees. And then eventually that preference kind of goes away as the infestation rate climbs. And so then they kind of transition to, oh, they're not on nurse bees as much, to, oh, they don't have a preference between the age of the bee. And Tropilaelaps, though, of course, doesn't feed on adult bees. So they don't really need to be on these adult bees. And you find them at very, very low phoretic mite levels. And so for my management trial that I did over there, is I think we had like 12 colonies, or that were my 12 or 13 control colonies. And I uncapped 200 cells in each colony. And I found like 575 mites at the end of my experiment in the brood.
Hmm.
I found 9 mites after doing 12 or 13 alcohol washes in all those
Wow.
colonies. So, and we're talking pretty high infestation levels. And so I also did a kind of dispersal. So I actually set up these interesting traps. They're called funnel traps. And there's a Medrzycki paper, I believe, from 2013, kind of explaining how these traps are. They're funnel traps, but what they are is that they are used to trap exiting bees. So essentially, you're trying to trap foraging bees, and you're trapping bees that are exiting the colony.
Hmm.
So I did that in the summer of when I was over there, and I trapped and washed and I alcohol washed them, and I was just looking for a single mite. I was like, can I find a mite on an exiting bee? Because then I can be like, okay, it's possible that if the mite is on an exiting bee, then it could go into a different colony on accident,
Right.
drift, and
Right.
then that's a way that it disperses.
It at least shows us a possible mechanism of dispersal. And it's amazing how little, for Varroa, when I was studying that in my own work, not all that recently, but not all that long ago, it was the same thing. I sat down and I said, well, what are all of the ways that a Varroa mite could spread between colonies? And very little research had actually been done. People had been focused on how to kill the things, but we hadn't really looked at their biology. So, we were confused about how they actually spread from one hive to the other.
Yeah, so I did these traps. I washed these adult bees. I washed probably a little over 7,000 bees on these exiting from 6 different colonies. I
Hmm.
found 0 mites. I
Hmm.
didn't find a single one. And then I kind of, but when I was doing my Tropilaelaps, I was doing brood inspections. I was checking the brood infestation. We had low brood infestation in these colonies. These colonies
Hmm.
had, probably less than overall on average one mite per 100 cells. So
Okay, so just not
if
that many mites overall.
you're just not that many mites. And so if
Hmm.
you have that low of infestation, they don't have a need to be on the adult bees, especially if they're not feeding on them.
Hmm.
So I went back over in January and we had colonies with a lot higher, I think average of about 25% infestation within these colonies. So about 25 mites per 100 cells. And for the first time, and this is a dispersal kind of note that I'm hoping to publish in the near future, is we actually did find mites on some of these exiting forager bees. So we did kind of prove that this is a potential method of dispersal. But the big problem is that we have this low infestation of less than 1%. We have this 25%, and I still didn't find a lot of mites on these
Hmm.
exiting bees. Most of them were coming just from a singular colony.
Hmm.
And so it kind of brings up the question, okay, well, what's the threshold that these mites are actually potentially voluntarily, theoretically getting on these adult bees
Hmm.
and then opening up the possibility? And that's why I think that similar to one of your papers that you have published is this becoming a large robbing behavior? And is that one thing that you're seeing strong bees from strong colonies come in and the mites are potentially able to more actively get on those bees and then be brought
Hmm.
back to these stronger colonies?
Got it. Well, and Varroa and Tropilaelaps share the same, quality in that they spend their whole lives living inside a pitch black honeybee colony, and so they've got to navigate by touch and by smell, and so they, like honeybees, are very sensitive to the smell of, is this a bee from the local colony or is this a bee from somewhere else? So it might not be that hard to imagine that Tropilaelaps has a system that lets them smell, ooh, that bee smells exotic and exciting. Why don't I jump onto her? Only to find out that, more often than not, that means that she'll be riding off with a robber and showing up in a brand new colony. So yeah, you're right, but it leads to that same consequence: is that the sicker the mites make the colony, the more likely the colony is to get robbed, and therefore the more opportunity they'll have to spread, which is the same thing that's true of, something like a waterborne human pathogen, right? If you get cholera and then you get diarrhea and you're pooping out all of these cholera spores, well, the more you poop, the more likely it is to get in someone else's water, and the more likely it is for somebody else to get cholera and somebody else to get cholera. But if it's easy to spread that way, there's no real consequence to killing your host. And so there's not much incentive for a varroa mite or a tropilaelaps or a cholera to get into a host and then be nice to it, to be gentle, to keep it, let it eke out a living, because they say, that'll be fine. We'll burn through these resources and then we'll go find somebody else's resources and exploit those.
And that's really the interesting thing, is how well are they actually able to identify that time point? Because when you see these heavily infested colonies, it is somewhat similar to Varroa, where you have so much brood disease, you have this
Hmm.
decaying brood in the cells. They're taking out all of their resources that they can actually feed on. They are eliminating the larvae and the pupae. There are no more, the bees are really struggling to take care of all this. The bees also somewhat almost retroactively, as it's too late, begin to develop a very high sense of hygienic behavior and begin to remove a lot of the brood. And I was seeing, like in some of these
Hmm.
heavily infested colonies, we're seeing almost a delayed response of, similar to what you might see in a Varroa, heavily infested Varroa colony, is it's like too little too late, but the bees are kind of recognizing, hey, we have a large problem here and we need to be
Yeah.
getting this brood out of our colony. So it's almost to the point where potentially it could be that these mites are clinging on to anything that they can, because they really have nowhere else to go, because they have nothing to feed on within the colony, because they burned through all those resources.
Right.
And it
There's
could
nothing
be
left.
essentially a last ditch effort. But you have to kind of think about, is these mites have to have created an environment bad enough for them that they no longer can substantiate themselves
Hmm.
and exist in that kind of environment to
Yeah.
begin wanting to disperse.
Yeah, well, and it's a risky maneuver, right? Any dispersal that any creature does, but certainly a parasite that has to be inside a honeybee colony that has brood for it to feed on, leaping onto a bee and saying, "Well, I hope I wind up in a honeybee colony at the end of the day," is a very risky move, but one that, potentially pays off for the few lucky ones that spread. Now, Varroa you only need one Varroa mite to get into a colony because they're all pregnant and they can all inbreed with each other, and they do that sort of by their nature. And so there's no real consequence to just having one of them go in and form a new colony. Is that also true for Tropilaelaps? Is there consequences to only having one mite, show up in a new hive?
So males usually only survive for 5 days, and females survive for roughly on average 50 days. So of course, if you had a male mite that latched onto a bee, it made itself, it's
Right.
not
No
nothing's really
big
going to
deal.
happen. Yeah.
Right.
So you're thinking that you really do need a gravid female, a female that is essentially pregnant and has the capability of laying eggs. But yes, then you can have that inbreeding effect. Now, Tropilaelaps doesn't lay just a single male egg followed by daughter mites. It's sometimes it can be more random where they
Mhm.
lay more randomly. It might be male, female, female male, or it might be male male male, or it's a little bit
Got
more
it.
random. It's about 50-50, as I believe was what the literature says, but there's not like the set progression like Varroa has.
Mhm.
And so you would probably only need that one female mite. And of course, this is your potential dispersal kind of mechanisms. Now, I'm going to hypothesize that beekeeper-mediated dispersal is probably much more likely to actually occur where you're moving a frame that has some Tropilaelaps mites on it or in the cells or in the brood. You're moving that frame to a new colony, and that's probably a very common way of
Mhm.
dispersal. But we're looking at, ways that, it could reach overseas essentially for us.
Right. Right. For something like Varroa, there's a great concern, looking at the arrival of Varroa in Australia, that almost certainly happened when a rogue swarm with mites came in and then sort of seeded the country with those mites. And so you can imagine the same thing happening with Tropilaelaps if they're able to sustain themselves on a swarm for a transatlantic or transpacific voyage. But if that doesn't happen, if they can't do that, then we can rule that out as a possible concern. Now we have to be worried about swarms, in an airplane or somebody smuggling brood, but maybe we don't have to worry as much about the other side. So that's just to explain to people why we waste our time doing all this research, is that it's actually quite important to know, especially even if it's just that one rare little instance where a mite can get over that country's border and get into a new place. We've got to know what the real risks are and what isn't a risk, so we know where we should be monitoring and where we should be trying to anticipate these invasions. So you said you've been working with the Apiary Inspectors of America, who are some of the people who are doing some of this work, trying to say, hey, here's a funny bug running around in a bee colony. If somebody takes a photo, that's going to get sent to the apiary inspector, or maybe to a university like Auburn or Cornell, or maybe someone like me at Betterbee, and they'll say, hey, what's this thing running around on my bees? And then we need to be able to recognize it. What's going to happen if somebody sends me a photo and says, hey, David, I'm here in Vermont and I found this weird mite on my bees, and I look at it and I send it to you and we send it to the USDA and we all agree that we're looking at a Tropilaelaps? What happens next? Hey, everybody. Sorry for the interruption. Since I interviewed Rogan, he's actually had more conversations with the USDA APHIS professionals who would be in charge of responding to a Tropilaelaps incursion. So, in his original answer to my question, he suggested that colonies in yards confirmed to have Tropilaelaps mites would be quarantined, and then hives in a wide radius around them would be tested for mites, and that infested hives may have to be burned. This is a really large worry for beekeepers. However, we've since learned that the planned APHIS response is state dependent, and they're going to treat things on a case by case basis. APHIS plans to evaluate any traces of Tropilaelaps individually, and then they're going to design a response that fits the circumstances. That means the response won't necessarily be wholesale burning of hives like we've seen in the past, like when Varroa got to Australia. Rogan asks that I cut out his now outdated response, but now we can rejoin the interview in progress and get back to what he and I were talking about.
But then could you do something to colonies, maybe 20 miles away instead of having a zone that's 50 miles where we're burning every colony in this zone?
Mm-hmm.
Is there something that, we can do as a treatment alternative that we're pretty sure it's going to kill every mite in that colony that the
Mm-hmm.
beekeeper can do where they no longer have to essentially char their colonies? And
Right.
so, because of course you find it, if it were to be found in California around almond pollination, now you have an enormous problem
Right.
of,
Especially if we
that's
find out that they're good at spreading from hive to hive.
Yeah, especially in that scenario.
Yeah.
And so part of my research over there was, can I do something that so brood breaks have been proven to be really effective. So these
Hmm.
mites don't feed on adult bees, and they actually are only able to exist for up to 3 days without feeding on larvae and pupae. Now, interestingly, as well is that larvae have to be 3.5 days old, according to the literature, before these mites begin feeding on the larvae.
There's not enough larvae there otherwise.
Yeah, essentially, if
Yeah.
it's a day one larvae, they're not really able to actively feed on that
Hmm.
larvae. And so the idea behind these brood breaks is that, well, if you eliminate all the brood within the colony, even if a mite emerged that day that you release the queen, she lays an egg. Well, it's going to take 3 days for that egg to hatch. It's going to take another 3.5 days before that larvae is old enough for that Tropilaelaps mite to actively feed on. Based on the literature saying that the mite dies within usually 3 days on the adult bee, that should essentially eliminate all of the mites within the colony.
So it's not just that they don't feed on the adults, it's that they can't feed on the adults. It's that you can starve them out as long as you can get rid of the right aged larvae, in a really effective and decisive way in that hive.
Essentially, yeah, if you eliminate the larvae, you eliminate the pupae because, of course, they can feed on the pupae within the brood. If you eliminate
Hmm.
all of that, you have zero brood in the colony. So essentially, you have like maybe it's a newly installed package or something like that.
Hmm.
Theoretically, those mites should not be able to survive. And that's one of the interesting things. So say a mite is able to survive on a plane. Well, it would need to be feeding on larvae relatively quickly.
Hmm.
Otherwise, it would most likely die.
Hmm.
And so we saw this, but there's been literature and there's been studies done that have done brood breaks. They get 99% efficacy, extremely good. They
Hmm.
don't get 100%. And think about it, that's what we're really aiming for in these quarantine zones because we want to
Right.
get rid of that
We have to
mite.
guarantee that there are no mites, not very few.
Mhm.
Yeah.
And so what the idea was behind my study is then I did a brood break for a part of my treatment group. I had a control treatment group, and then I did a brood break with an additional chemical treatment, one being oxalic acid and another being formic acid. So
Hmm.
I ran this experiment essentially for 60 days, and I caged the queen for 24 of those 60 days
Hmm.
because, a drone is going to take 24. So if the queen
Right.
laid an egg, 24 days later, that drone will have emerged. And
Right.
so I should have no brood within the colony.
Hmm.
And so then I came back and did sticky boards and brood assessments. And so brood assessments are generally a lot of times what you do to actually assess Tropilaelaps infestation because you can't do an alcohol wash. So you're
Hmm.
mainly doing brood cell
Yep.
infestation.
Just opening up cells, counting how many
Mhm.
mites you find, or how many have any mites inside.
And I found zero mites on day 50 in my brood break plus formic acid colonies on the sticky boards,
Uh-huh.
zero mites on day 60 in my brood cell infestations, but I found a few mites on day 60 for my sticky boards.
Hmm.
And so the problem is that of course I housed all of these colonies in the same location. And now we kind of know that they
Right.
could have been
Maybe those are a couple of mites that were recent
ex
imports from somebody next door.
exactly. And
Got it.
so what we're hoping to do is that's one of the things that I want to go back over and actually study. In this upcoming years, I'll be going back over in August, and the goal is to kind of recreate this experiment, but have yards where we do all the colonies in that yard get a brood break plus a formic acid, and then we have another yard where some of the colonies get that, but we also have one or two colonies that aren't receiving any treatment. And then we're seeing if in the yards that every colony gets the same treatment, we can't find any mites in those colonies.
Got
And
it.
then unfortunately, we can never really 100% say, hey, we've eliminated all the mites. We
Hmm.
can't with 100% certainty say that, but that's the idea of what we're trying to do because that would then provide an alternative. So say we get this, well, our quarantine zone for burning colonies becomes smaller. We say, hey, everybody that's in this zone, you now
Hmm.
have to brood break and you have
Yep.
to do a formic acid treatment.
Right. And don't move those bees for the next 2 years, and we're
Yes.
going to keep a close eye on them. But
Hmm.
we think we've got a method that we can hit them hard enough with this sledgehammer that they're not going anywhere without having to exterminate the bees. Well, this is the whole point of these is to talk about science and exciting new stuff, not to sell products. But I will just point out that if someone was interested in brood breaks, Betterbee does sell these. I grab it lives next to my desk because I periodically hold it up in about half of the Zoom calls I do. But this is a queen excluder walled cage, which is perfect for somebody who wanted to cage a queen and create a brood break, because you can let the queen lay eggs, then at the certain appropriate time pull that frame out and throw it in the freezer. So basically all of the brood was on that frame, and now you've removed it. So there's maybe some potential if Tropilaelaps did come to the United States. If that worst case scenario comes true, then maybe we don't have to become Thai beekeepers who only have 7 frames of brood and 3 frames of honey in our single box hives. Maybe there would be a strategy based on your research using tools like that would allow people to allow the bee population to grow, and then impose a brood break like this to purge the Tropilaelaps from the apiary, and then let them start growing again. And sure, maybe there might be a couple of mites here or there, but they're manageable as long as scientists like you actually develop the tools and the techniques that we need.
Yeah. Yeah, and so that's really what our goal is to really come up with different management strategies, try them in different environments. We're hoping to run a study this year. One of my colleagues who actually just defended his PhD and graduated and sticking on as a postdoc at Auburn University, he's going over to South Korea, and he's hoping to do some of this research and management research over there to determine in a more temperate climate, not quite as hot as what we're dealing with constantly in Thailand, are these same management treatments really effective?
Got it. Well, and at the same time, if these mites are so dependent upon having brood, I'm up here, I'm next to Vermont, I'm here in upstate New York. I have a pretty long period of time where I don't have much brood to speak of in my hives. And I would dare say that for most of the time in most winters, I've got 0 brood in my hives for certainly more than 3.5 days. So does that mean that if Tropilaelaps showed up, dummies like you living in Alabama are going to have to be fighting them, but I'm going to have an easy time? Is that what we're thinking could happen, that it'll be a lot easier in these cold winter climates?
I can't say anything for certain, but I would bet money that it is going to be a bigger problem, and it's going to be, our colleagues and our friends down in Florida, in that honeybee lab are going to
Mm-hmm.
be the hardest hit most likely, because they have some of the most brood around year round. And this is an interesting plug. So you plugged one of your queen cages. I have to
Oh, yeah.
plug one of our cool, interesting studies. So at Auburn University, we've been partnering with some different universities, especially throughout the Southeast, but ranging all the way up to Cornell as well, having this brood monitoring kind of thing that we've been doing throughout the past 2 years. And so the interesting thing and idea behind this was my advisor, Dr. Geoff Williams. He went into his colonies in mid-November after kind of a cold thing snap happened in Auburn, and realized that he had extremely low, almost no brood in late November.
Mm-hmm.
And then he was like, oh, maybe we have less brood than I thought. And essentially that was like his low point of brood throughout the entire year. And so what we're doing is we're essentially having about 10 colonies per group, and we're just going out every 2 weeks and categorizing how much to cap brood. Because this is important for Tropilaelaps, but it's also interesting for Varroa mites, is if
Right.
you can get in, do an oxalic acid dribble when you have your lowest amount of brood, it's going to be the most effective.
Mm-hmm.
And then you might get away with not having to treat again in the spring. And so you can make it all the way till, June, July, August without another treatment if you can get in during that time point. And so that's one of the interesting things. So thinking about it in the Tropilaelaps point of view, and from anecdotally what we've somewhat heard from South Korea, is they still have large problems with Varroa. Tropilaelaps is not the mite that is causing them as much issues, and they have more areas where they have not potentially full brood breaks, but more of the kind of maybe baseball, softball size clusters
Right.
that we
Very
see.
small amounts of brood.
Mhm.
Got it.
And potentially that is allowing the Tropilaelaps mites to subsist and continue to survive, but it's still causing Varroa to kind of be a predominant issue in their beekeeping compared to the areas that I visited in Thailand, which are having fairly high levels of brood year round. They're going to almost always have these problems with
Right.
Tropilaelaps
Well,
mites.
much like our colleagues, in Gainesville, or you guys,
Mhm.
if you leave a sick colony or a small colony unattended for a week, you might have small hive beetles come in and overwhelm the whole thing. Whereas up here, I have small hive beetles. I have
Mhm.
to deal with small hive beetles. They're occasionally going to cause some problems for me, but they're not a horrible plague just because keeping bees in a different climate means that some pests are worse and some pests are better. And I guess the context is important for beekeepers, especially folks who have started beekeeping in the last, couple of years or couple of decades, is, there were no mites to speak of on honeybees in the United States. And then in 1984, tracheal mites showed up, and they were a catastrophe. They were a disaster. Lots of bees died. The tracheal mites were causing all sorts of problems. People were desperate for treatments. And then in 1987, Varroa mites arrived in the United States, and they were also a disaster, and they
Mhm.
were also a catastrophe. Now we hardly ever talk about tracheal mites. We know that Varroa are a serious problem, but people who pay attention and really learn Varroa biology can generally keep their mite levels at a low and manageable level in the US. And so
Mhm.
if Tropilaelaps did ever get to the US, which isn't a guarantee, and if Tropilaelaps got to the US and then got established, right, those containment zones didn't work and they do start spreading around the country and into bee populations, it, it's entirely possible that they'll just be another annoying parasitic mite that beekeepers have to learn how to manage. But it's not going to be the apocalypse that some people like to imagine. That if a single Tropilaelaps mite gets into the U.S., we might as well just burn all the hives and throw ourselves into the ocean because nothing will ever be the same again. It's maybe not going to be quite that bad.
Yeah, and so that's a very common question is, okay, we have all this interesting things about Tropilaelaps. We're starting to learn more. We still have a lot of unfortunately unanswered questions that we'd like to know more about. But one of the most common questions that I often get is, okay, when's the mite going to get here?
Hmm.
And that is one question that I, of course, cannot answer. I tell beekeepers it could be within 5 years, it could be 50 years. I
Right.
don't know. The thing that we want to be is we want to be prepared. It does seem like it's expanded its geographical range more in it seems like the past, decade to half decade than it had in the previous, 30 plus years
Hmm.
or 40 years that it had kind of been in honeybee colonies. Because this isn't a new parasite to areas like Thailand. They've been
Hmm.
essentially dealing with this parasitic mite since they've been beekeeping. And that's
Beekeeping
why,
in Thailand is beekeeping with Tropilaelaps. That's just part of it.
exactly.
Hmm.
And so they've managed to just kind of coexist with the mite. And it's similar to Varroa is you do your mite treatments and you go about your day. And if you are a good beekeeper and you're staying on top of things, you're able to handle them.
Hmm.
And so that's the question is I can't tell you when the mite would get here, but I do think that we are hopefully developing these strategies so that when it
Hmm.
gets here, we have things that we can talk to beekeepers about and we can implore them, hey, do this and this and this. And it's just going to be another problem that you're going to deal with. I don't know if it's going to be the catastrophic that, like you said, we're going to have to burn all our colonies and pack it up and I'm going to have to find a new career.
Right, you and me both,
But
brother.
I don't know if it's that bad, but I mean there are dangers of it, and it is scary to see that this is something that reproduces faster than Varroa and can kind of grab its hold on colonies more quickly than Varroa. And so there are scary aspects to it,
Hmm.
but I think there are hopefully developing tools that we can say, okay, if you manage your bees correctly, we can continue to combat this pest if it
Hmm.
ever were to reach the US.
Right, and it's as you've said today, it's faster than Varroa. It's nastier than Varroa. It makes more bite wounds, and it can do all this stuff. But in some ways, it's a little bit weaker than Varroa because it
Mhm.
does rely on that brood of just the right age. They're picky eaters, and that could be an advantage to us because we as beekeepers we know that we can manage the brood. Right, we can make there be more, we can make there be less, we can time it, we can adjust those sorts of things, and maybe we just need to get smarter about managing our bees or helping our bees manage themselves in order to fight these should they ever show up. So Am I correct in saying that your official answer to the question of whether the doom and gloom is appropriate is that concern is appropriate, certainly more research on these is appropriate and critical so that we know what's happening if they do show up, but that we probably shouldn't just be giving up beekeeping, that it's not unethical for me to convince somebody to pick up beekeeping as a hobby because it's all just going to be for nothing in 2 years when the Tropilaelaps mites ruin it all.
I would say that that somewhat accurately summarizes it all up into a little bit more of a comprehensible manner.
Right.
Yes.
Well, Rogan, it has been a great pleasure talking to you. I don't want this video to go forever, so I think maybe we'll end here. I look forward to having you on again when you've done more exciting research and you've got more stuff to share. But it's so valuable for me to be able to talk to other scientists to get this really important research out in a way that beekeepers can get it. I'm so glad that you're publishing your papers. It's critical for your research. It's critical for other scientists, our colleagues, to read and be able to evaluate this stuff. But a lot of beekeepers are not sitting down and reading all of the publications that come out of the bee labs. So it's really valuable that you've been able to take some time here and share some of your insights, because I think it will help more beekeepers understand that this is a real threat. This is a real concern. It's not just an apocalyptic boogeyman, but it's also not fictional. It's a real thing that real scientists are doing real work on to try to help us get prepared to fight them if we ever have to do so. So thank you again for coming and sharing your knowledge with us.
Yeah, well, thank you so much for providing me with the opportunity to actually reach, some of the beekeepers that I wouldn't normally be able to reach with some of my research and things like that. So this is just a great series that you're putting on, and I'm really appreciative of you giving me this chance. Thank you.
Yeah, thank you so much. All right, see you next time. Okay, excellent. All right, so we're already recording, so I'll just jump into it, and then we'll be into it. Except I've lost my pencil already. What on earth did I do? Well, I don't need it. All right, I won't write any additional notes. That's so weird. I'm sure I'll find it eventually. All right, well, as you can see, Rogan, I'm losing my mind. Let's jump into things. Okay.